Okay, welcome biology students. This is Mr. Howard, and the purpose of this lab is to walk you through the Topic 3B Catalase Enzyme Lab. I'm going to go through a few of the directions and the procedures and just kind of walk you through what those procedures look like. First step on the procedure says wear goggles, so make sure you got your goggles in hand and put those on. Step 2, connect the rubber stopper and plastic tubing. So these have little spin connections, just spin those on so they're nice and tight, and to connect that also to the gas pressure sensor. Make sure they're nice and secure. Step 3, connect the gas pressure sensor and the LabQuest. I've already done that. You can see that that's plugged into channel 1. We'll go to the LabQuest app, make sure that we are on the right units here. 98 or 100 is approximately the atmospheric pressure, so that'd be an appropriate reading at this point while we have our rubber stopper hooked up to the gas pressure sensor and the LabQuest 2. So the next step is to set up your units and collection techniques. So we're going to look at the rate. I'm going to change that to 0.5 samples per second. So click up here on the 1 and put 0.5. So every half second we're going to take a reading. Click done. The interval is at 2. The duration, we want this to run for 180 seconds. So this one is currently at 900, which is way too long. Make sure you set this up so your LabQuest automatically stops collecting data when you want it to. So duration's 180, that's done. Step 5 says place 4 test tubes in a rack. Here you have 4 test tubes that work really well with the rubber stopper that we have connected to the gas pressure sensor. The next step would be to take our H2O2 or hydrogen peroxide. This is a 3% concentration, the same concentration as hydrogen peroxide you might find around your house. So it's relatively safe, nothing to be too concerned about here. You need to fill up 6 mils into each one of these test tubes. On this pipette, I've marked with a yellow piece of tape, filling up with liquid. This whole part here, this whole tube, is approximately 1 milliliter. So if you get 6 of these and put that into your test tube, so you can see the amount of liquid in here. The most important part, it does say on your lab approximately 1 inch. It actually looks like a little bit more than that. The most important part for this next part is to make sure that all 4 test tubes have equal amounts of liquid in them. You've already stopped and read 7 through 12, so you already know what to expect when we do this. The reason we had you read step 7 through 12 is because they're kind of time sensitive. Once we add in the yeast to these test tubes with H2O2, we're going to need to connect the rubber stopper fairly quick. So using our dropper bottle labeled yeast, for demonstration purposes, this is just water in here. If this was a yeast, you'd want to make sure to check and see if there's any on the bottom. It almost looks like sand, and then it might just be a more clear, cloudy liquid above. So you want to make sure you give this a nice little shake to mix around the yeast. And then you want to make sure that you plunger out any liquid that might be in the dropper. Make sure that's emptied before you pull from there. Make sure that that drops all the way to the bottom and hits the H2O2 and doesn't fall on the sides. As soon as we get that entered in there, give this a little bit of a shake and immediately add our gas pressure sensor firmly, but not too hard because we don't want to crack this. Once you have that on there, you're going to want to click play. That'll begin graphing your data. Run number 1 that we just finished. You're going to click the file cabinet Let's get change over to run number 2. Now we're ready for the second test tube.